Hey everyone! Today I'm going to explain and outline my main project for this summer. Here's my main goal. I want to see if patients who have had COVID-19 and recovered have antibodies to other coronaviruses they could not have come into contact with because they are involved with other species. So, how is this going to work? If you've seen my lab tour video, you will know some of the lab instruments I use and their main functions. What I'm going to do first is coat parts of a 96 well microtiter plate with different coronaviruses. Those that COVID-19 patients likely could not have come into contact with because they affect other species. I have 5 different coronaviruses, which I'm going to call A, B, C, D, and E. Under the right conditions, I can attach these viruses or parts of them to the polystyrene plastic of the microtiter plate. After I coat the plate with virus, I am going to wash off any virus parts that did not bind to the plate. After I wash it, I'm going to block this plate with a blocker to keep patient samples or any antibodies from binding to the plastic sides of the plate. I only want specific binding between the sample and the virus. After blocking the plate, I will wash again before pipetting patient samples into the plate. These samples are from recovered COVID-19 patients and are from the local blood center. They have no infectious virus in them and are safe. I will then incubate the plate that has the virus and the sample in an incubator for 60 minutes at body temperature. I have to incubate the plate at 37 degrees Celsius in order to be able to detect the antibodies I need to see. After the plate is incubated, I will wash off the patient sample with the automated washer so that all that is left is patient antibodies bound to the virus, if they are there. The next step is to pipette a conjugate antibody into each well. The conjugate antibody detects the human antibody with an enzyme called HRP attached to it. There you can see the conjugate antibody and the patient antibody. So that little circle you see on the end of the conjugate antibody is HRP, which stands for horseradish peroxidase. So, after this antibody is attached, the plate must undergo another wash sequence. We don't want any extra non-specific binding. We just want conjugate antibody to bind to the patient antibody. Okay, so finally it is ready for TMB. So let me quick re-show you what we have on the plate. So we have the plate, we have our patient antibody, we have our conjugate antibody, we have our HRP. And so, yeah, now we are going to put in TMB. So when I pipette TMB into a well in which there's antibody bound to the virus, the well will begin to turn blue. By using the scanner, I can read the absorbance of the blue color, or I can stop the reaction on the plate with stop solution and read the absorbance of a yellow color. So, as you can see where I put acid, that's another way to say stop solution. So the readings from the scanner will tell me whether or not patients who have had COVID-19 have antibodies to other coronaviruses they could not have come into contact with. If they do, it would indicate cross-reactivity between species. So after I've used this scanner, I will learn a lot of important information.